Bacteria are one of the types of microbes which can only be seen under a microscope. They are classified into 5 main groups according to their shapes: cocci, bacilli, spirilla, vibrio, and spirochetes. Can you imagine how many bacteria are there on Earth? Animals contribute a total of 2 gigatons of carbon, while all bacteria combined contributes a total of 70 gigatons of carbon on Earth. 1 teaspoon of soil can consist about 1 billion microorganisms, which is same as the total human population in Africa. And within your gut, the total bacteria can weigh about 1 kilogram. Bacteria is usually detected by plate culturing in the laboratory. They can form colonies on the agar plate. The number of bacteria is calculated by counting the colony-forming unit on the plate. However, there are some bacteria that cannot be cultivated on plate. These bacteria are called viable but non-culturable bacteria, also known as Vbnc. Vbnc bacteria can neither grow nor divide. Why? Because when they are under certain environmental stress, they enter a viable but non-culturable state to fight for their survival, such as when they face starvation, face extreme temperature, exposure to antibiotics, or chemicals. Vbnc was first reported in 1982. With the understanding of such a switch into Vbnc state, the total amount of bacteria reported by plate culturing is always underestimated. Flow cytometry provides an easy-to-use and cost-effective test system to quickly enumerate bacteria in many types of water-based samples and beverage-based samples. Water-based samples include those from industrial water, aquaculture facilities, and environmental sources. Beverage-based samples are wine, bottled water, and juice that include packaged juice and juice drinks. The principle that flow cytometry applies to quantify bacteria is by a membrane-permeable dye called cystine green. Cystine green can enter both live and dead bacteria and label the bacterial DNA. Flow cytometry reports the amount of detected cystine green as the total cell count. To quantify dead bacteria, another dye called cystine red is used. Cystine red is a membrane-impermeable dye that can only enter dead bacteria with loss of membrane integrity but cannot enter live bacteria with an intact membrane. By measuring both cystine green and cystine red inside the bacteria, flow cytometry can distinguish live and dead bacteria and report the number of live bacteria as viable cell count. With the ability to detect bacteria in the water samples by flow cytometry, a highly sensitive monitoring system can now routinely report bacterial cell counts. If there is an abnormal increase in the total cell level, flow cytometry can quickly capture such change and create an alert. Flow cytometry is also able to report cell counts with high nucleic acid and low nucleic acid content in the water sample. This HNA and LNA bacterial content is called the fingerprint. Even if there are no abnormal changes in the total bacteria level, flow cytometry can detect the change in the fingerprint in terms of the HNA and LNA content. Switzerland is the first country that has adopted flow cytometry to quantify bacteria in the water supply. Nowadays, with the online water monitoring system, water sampling, bacterial staining, and reporting can all be done automatically and 24/7. Thanks to this highly automatic and robust water monitoring system, the water is safe for consumption.